I'm Daniel. I am from the Czech Technical University, and today I will present my work on the scatterplot visualization of hierarchically clustered data. I have designed an application for interactive visual analysis of large datasets of medical measurements. The data consists of hundreds of thousands of points. The points are in a high-dimensional space and are hierarchically clustered. Visualization of such data help with insight and understanding of underlying biological processes, which is important for drug development, disease diagnosis and prediction, and other medical applications. Each point in the datasets has a category, a position in high-dimensional space, and a position in two-dimensional space. The position in two-dimensional space is obtained from the high-dimensional position using a dimension reduction technique. The points are subdivided into clusters by their category. Overlaps of clusters of different categories signifies interactions. So this is an important feature of the dataset that we want to focus on with the visualization. The points are then subdivided into a hierarchy of clusters by their proximity. Unfortunately, the data available to us do not contain this hierarchy, so we have to subdivide the points ourselves using the k-means ++ method. So these are the two datasets available to us. We see a number of points, number of categories, and the original dimensionality of the points. The points are drawn on the images with their 2D position, and each color is for one category. Our visualization aids with the tasks of cluster identification, density estimation, and overlap identification. I will now briefly explain how the visualization helps with these tasks, and then I will follow up with more detailed explanation. So, cluster identification requires coloring the clusters with distinctive colors, but there is a lot of different clusters. So we have used the technique of the dynamic color palette developed at the UVin that assigns distinctive colors only to the clusters that are currently visible on the screen. Next, we have the density estimation. On the middle picture, we can see varying densities inside those two blue large clusters in the top left of the middle image. And lastly, there is overlap identification. There is a yellow cluster in the last image that wasn't seen on the previous images. This is because it was completely overlapped by the blue clusters. So let's start with the cluster identification. The dynamic color palette allocates colors in the HCl color space. The hue ring is subdivided into number of wedges, each wedge for one top-level cluster. Top-level cluster is the one defined by their category. Color wedge, this is a range in the hue space. We have displayed these wedges in the top image. So the size of the wedges is given by number of points visible on the screen of an associated cluster. So as we zoom in, some clusters leave the visualization, their points leave the visualization, causing the hue wedges to shrink while the others grow. So this allows us to subdivide the grown wedges and allocate distinctive colors to subclusters. We can do this repeatedly, creating a hierarchy of wedges that mirrors the hierarchy of clusters. So on the bottom image, we have a zooming process on the cluster marked by the red circle. So at first, we have one color wedge, and the color assigned to the cluster is middle of the wedge, so that's the black point. So we zoomed in on the middle part. Now the color wedge has grown, and it was subdivided into 3 different wedges. We can also see that the cluster is now colored with 3 different colors. Now we zoom up again on the green part, and in the last part, we now see that the green part is also colored with now different colors. There is now also more color wedges. But hue alone was not enough to distinguish the colors, so we have used additionally the luminance channel. Luminance is assigned in 5 fixed levels of luminance assigned to top-level clusters. The subclusters then inherit the luminance level of their parent. So now we have density estimation. Visualization of density requires drawing the points with transparency. On the left image, points are drawn without transparency. On the right one, with transparency. We can see a difference between those two pictures in the shape and density of the blue clusters in the red circle. Some clusters have a majority of their points focused in small area, while the rest are outliers. This is also seen when we use density visualization. So in the left image, the red cluster appears to be quite homogeneous, while in the right picture, we see some focused area. That's this one. Unfortunately, we cannot use luminance channel together with transparency because darker clusters will only appear to be sparser. Therefore, we have created two separate modes of visualization. One focused on the cluster discernibility that uses the luminance channel, and another one focused on density that uses driving points with transparency. The density of clusters varies strongly. This causes the sparse areas to be poorly visible, while in the dense areas we cannot distinguish the densities. Therefore, we have used rendering in two passes. In the first pass, all points are drawn in a way that there is at most one point drawn in each pixel of the screen. This is ensured with the stencil test. In the second pass, we draw all points without the stencil test, this time with higher transparency setting, so we could still distinguish even those dense areas. So on the left image, we have a single pass rendering and high transparency settings. The sparse parts are not visible, unlike in the other two pictures, but we can discern the densities in the red circle. On the second image, we are again using single pass rendering, this time with lower transparency settings. The sparse areas are now visible, but the area in the red circle now appears to be homogeneous. And in the last image, we are using two passes. We can see both the sparse areas, and we can discern the densities inside the red circle. So when we don't use transparency, clusters are overdrawn with clusters that are drawn after them. Since we are drawing with OpenGL, we can solve this by enabling the depth test and assigning random depth to each point. This is similar to random color dithering. So in the left image, there is an orange cluster in the red circle. In the middle image, there is a purple cluster. All we did change between those two images that we reversed the rendering order. So we see that without depth test, the differences can be quite big. In the last image, we are using random point depth. We can now see both of the clusters, the orange one and the purple one. The distribution of points now corresponds to the distribution of the densities of the clusters. But still, some clusters are very sparse and they will be still completely covered by a dense clusters. Therefore, the user is given the option to select clusters. This can be done either by clicking directly on the cluster or clicking in a node of the hierarchy visualization that was added for this purpose. The selected cluster will be drawn over the other clusters, and at the same time, the other clusters will be drawn darker so that the selected one would stand out. So in the left image, we see in the red circle parts of a yellow cluster, and after we select it, we can see now that the cluster overlaps all over the blue area. So now we have video. I can run the video now. So now we have video where we demonstrate zooming on different clusters. After zoom up, we can see that the clusters are now colored with different colors and are colored up to subclusters. We can also notice that colors will change when we move the view. So now we see the change in colors as we move in, because the color wedges are reallocated to different clusters. So now we have video where we, okay. So in this video, we demonstrate the results when selecting clusters. Now we select a group clusters of same area. The clusters have similar distribution of densities. We can see that they form such a sparse, narrow corridor that is bounded by walls of higher density. So in conclusion, we have achieved application with the real-time rendering. This gives advantage over other visualization that they do not use GPU acceleration, and the visualization takes even minutes to draw. We have also used hierarchical coloring that allows to distinguish the hierarchy of the clustering and also improves the discernibility of clusters. The application also have modes of visualization of density and overlaps. I have continued my work since I wrote my article, and I added more visualization options. The points can also be drawn after conversion to a density function. The density function can be either contoured, we see this on the left image, where we draw an isoline of a constant density. We can also perform relief shading on the density function, which gives nice 3D impressions of the cluster's density, and it also helps visualizing overlaps. So that's all for me. Thank you for your attention.
Thank you very much. Are there any questions?
So this might be a bit random, but where did you get these data points? Or did you define some functions and edit some noise? Or is this from some real-life phenomena? Or can you expand on that?
These are real measured data. They were provided to us from, I think, Charles University by some researcher.
Really interesting, thank you. And did you try your technique on other data, or like or was this the only one that you tried?
Well, at the beginning, I tried to generate some data, but it's difficult to generate some data that are so various and interesting like the real ones. So it's much better to do it on real data.
Yeah, thank you, because it looks great, and it's really, really colorful, and it looks nice. Thank you.
Other questions? Victor?
Okay. You can think of a question.
Okay, so I have a question. So the clustering that you perform, so you perform it in the original higher dimensional space, or how is the clustering performed on the data?
The clustering is performed in the two-dimensional space.
And so what kind of algorithm do you use to perform the clustering? Because the clusters seem kind of like,
Sharp
it looked like
boundaries,
difficult.
Well, they would be like more, the clusters would be less, the borders would be less determinate if you used the clustering in the original high dimensional space. That's true. That would be probably better.
Yes, thank you.
Anybody else? Maybe I have a question. So you mentioned these are real measurements. From like which domain are they?
We were not told what the data actually represents, but I was able to infer something. It's probably something like protein interactions. Mm-hmm. I looked up some of the headers from the tables that we are given, and one of the information is antidote in human bloodstream.
Mm-hmm. All right. If there are no further questions, let's thank the speaker. And yeah, thank you very much.